Welcome to the video for chapter 13.3. So in 13.2, we talked about the physical agents of disinfection. In chapter 13.3, we're going to talk about the chemical agents of disinfection. So let's get on to it. But before we start into the lecture, let's do a couple of review questions. So review question number 1 states, complete destruction of all microorganisms on a surface can be achieved by A, antisepsis, B, disinfection, C, sterilization, or desanitization. Okay, so take a second, pause the video, and try to figure out what the answer to this question is. Okay, so review question number 1 is a vocabulary question. So this is knowing what these different terms mean. Okay, and the question says that we want to destroy all microorganisms. So out of these 4 terms, there's only one of these that destroys all microorganisms, and that is C, sterilization. This is the only possible answer here. It's a good question to know. Okay, so review question number 2, which is also a good one to know, states, you were tasked with sterilizing an injectable drug for use in human patients. Which method would you choose if the drug were heat sensitive? Would you choose A, autoclaving, B, treatment with phenolics, C, filtration, or D, dilution in 90% ethanol? Okay, so pause the video again and see if you can't find the answer for this one. Okay, so if we were looking at this question, and we've done 13.1 and 13.2, there's 2 of these answers that we can eliminate by the sheer fact that we haven't talked about them at all. Okay, so we can eliminate B, treatment with phenolics. Okay, this is not something we've talked about. And we can also eliminate D, dilution in 90% ethanol. So that means that A and C are the only possible answers for this question. Now remember, the question says that the drug is heat sensitive. Well, if it's heat sensitive, we don't want to use A, right? Autoclaves use high pressure to get high temperatures. Okay, so the answer for this is C, filtration. So we'd use one of those syringe filters that we saw in 13.2. Okay, so 13.3 is all about chemical agents of disinfection. Okay, so you are going to be able to list different factors that can affect the efficacy of a disinfectant. Okay, you're going to be able to explain how disinfectants are compared. Okay, you need to be able to list the basic forms of commercial disinfectants and their various uses. And you need to describe the emergence of microbial resistance to disinfectants. So one of the first things we need to talk about are the different factors that can limit how useful a chemical agent is. Okay, so how useful a chemical agent is going to depend on 4 things. Well, 6 things. Okay, so the first is that organic load that we talked about in 13.1. So how much organic material is there on the area you want to disinfect? Okay, so things like blood, feces, vomit, they're actually going to interact with the chemical agent and they actually can bind to the agent, which means it's not available to actually kill bacteria. Okay, so a lot of times when we're disinfecting a surface, we have to clean it first, get rid of all that organic material, then use the disinfectant. Okay, chemical agents and disinfectants are also going to be dependent on the type of microbe present. Okay, so ideal disinfectants are going to have a broad range of activity, meaning they're going to kill a broad range of microbes. So depending on how much you want to spend and whether it's commercially available, this is going to dictate how broad your range is. Okay, so when we're talking about chemical agents, we have to talk about how corrosive these agents are. So when we're using these chemical agents, these agents of disinfection, we need to make sure that they're not going to corrode any of the surfaces. Like if it's metal, is it going to cause it to rust? Is it going to damage our skin and start to strip our skin off of our hands or arms? Because these levels of corrosiveness make it so that the chemical agent is not useful, right? You don't want to cause damage to our cells when we're trying to clean something. Okay, so one of the other things that we need to consider is stability. Okay, we want something that after we open the bottle and we go and use it to clean something, that it will still be useful 2, 3, 4 weeks, 6 months later. So things that you open and they become unstable very quickly, they're not going to be useful. So actually, these things we don't want to have on the shelves, because if somebody buys a chemical and then leaves it on their shelves open for a few months and it only lasts a couple of weeks, what can happen is that they can come back and use that cleaner and think they're actually cleaning a surface when it's not doing anything at all. So stability is a really huge factor about how useful a chemical agent is. We want them the most stable we can get. Okay, how it smells is also going to affect how useful a chemical agent is. If you cannot stand to clean with that agent, you're not going to clean with it. So it doesn't matter how good of a cleaner it is. If it smells bad, you're not going to use it. Okay, and then the final one that we need to talk about is surface tension. So this is how water pools together. And if you remember from Biology 201, we did the surface tension on a penny lab, and we got those bubbles to be pretty big and overlapping the sides of those pennies. Okay, that's a substance that has high surface tension. So high surface tension is going to bead together, kind of like water on a waxed car. Now, for disinfectants and chemical agents, we actually want low surface tension. We don't want high. Okay, we want low surface tension so it doesn't stick together, and that will allow the disinfectant to penetrate into cracks because it's not sticking together like it would if it had high surface tension. Okay, these are the ways that we deem if a chemical agent is useful. Now, how well does that disinfectant work? The CDC actually has some tests that they run, and they use it to classify cleaners by their level of disinfection. Okay, so we're going to have some cleaners that have high levels of disinfection. Okay, they're going to kill things like vegetative cells. Okay, these are just normal cells going through normal growth. Mycobacterium, spores, so endospores, fungi, and viruses. Okay, so something with a high level of disinfection is going to be able to kill and get rid of all five of these things. Okay, these are going to be things like ethylene oxide, formaldehyde. Um, but the problem with things that are high levels of disinfection, um, classified by the CDC, a lot of these are toxic. So they're only used in certain conditions and with a lot of safety measures involved. Okay, we have in the intermediate level, this is going to be things like phenolics and halogens, so things like bleach. Okay, so we can see here they're going to affect everything but spores. Now, things that have low levels of disinfection are going to be things that affect normal cells, maybe some fungi, and maybe some viruses. So they're not going to touch our mycobacterium or our spores. Okay, so the way that we classify things by their level of disinfection is we use what's called the use dilution test. Okay, so this is a test that's going to test, so it's going to determine how well a disinfectant kills microbes that have been dried onto a surface. So we're going to test both porous and non-porous surfaces. Okay, so if we look here in the um, in the pictures, these examples, we see these metal stainless steel little cylinders that we have here. Okay, this is going to be our non-porous surface. Okay, and if we see down here, we see this white thing. This would be a tile, so it's a porous surface. So these are the two things that we're going to test. So what we're going to do is we're going to arrange them on, say, a petri dish. And what we're going to do is we're going to apply microbes to them, and we're going to make sure the microbes drive to the surface. And then what we're going to do is we're going to use the disinfectant on them. Okay, so once the disinfectant is used, we're going to then take these cylinders and we're going to put them into a test tube with broth culture, and then we're going to put them in the incubator. They're going to be capped, and we're going to see if anything grows in this broth. Okay, so depending on how much growth you're going to get is going to dictate the level of disinfection you have. Okay, so some of these are not going to have any growth, while in some of them, they might have some growth. So like the intermediate, so the bacteria that causes endospores, we're still going to get growth with that one. Okay, so for low levels of disinfection, we are going to see growth on the mycobacteria and spores samples. Okay, so we also have lots of commercial disinfectants. So these are things like ethanols and iodines and chlorines. Okay, so some versions of bleach actually fall underneath commercial disinfectants, and these are going to be low-level disinfectors as classified by the CDC. Okay, all of these disinfectants are going to do pretty similar things. What they're going to do is they're going to disrupt the parts of the cell membrane of these microbes. Okay, they might damage the DNA, they might denature proteins, and they might degrade lipids. Okay, so it all depends on the disinfectant that you're using. Okay, the thing is with disinfectants, the more parts of the cell that they affect, okay, the less likely a bacteria cell can evolve resistance to them. Which is just like antibiotics, bacteria can grow resistant to certain cleaners. Okay, so besides using chemical disinfectants, we're starting to look at other things that we can use. So things like metals, things like copper, silver, antimony, they have natural antimicrobial properties based on their molecular structure. Okay, so we can make things out of these metals and they're going to have clean, sterile surfaces. They're going to act as their own disinfectants. Now, these are going to be something that are going to be ideal to use in high touch surfaces. So like the counter in a doctor's office or the rails of a hospital bed. Okay, things that are exposed to pathogens all the time, but we need them to be antimicrobial. Okay, so those are some of our commercial disinfectants. That is it for chapter 13.3. We will see you in chapter 13.4.